I work in imaging, predominantly magnetic resonance imaging, and we've been developing a technique over the last nearly 10 years now, which is a metabolic imaging technique. So we work in magnetic resonance. Magnetic resonance is a very insensitive method, and we've been working on a technique that increases sensitivity by about 10,000-fold or more. And it's allowing us to image in a way that we've never really been able to do before. So we're able to look at metabolites, small molecules inside cells which are involved in energy production in cells, and we can monitor that process in cancer cells. Cancer cells are very good at taking up certain molecules and making energy and growing very fast, and so we're able to image that process. We've really focused mainly on looking at treatment response. So we know that patients can respond very differently to the same drug, reflecting the genetic makeup of their tumours. And so one way to find out which is the best drug for a particular patient is to treat that patient with the drug and now ask very quickly, possibly within 24 or 48 hours, whether the drug's actually doing anything in that patient's tumour. And then you can then select that drug and carry on treating, for example, or if it's not working, possibly change. So what I was talking about here was actually brain tumours. I was in the session working, and it's not an area we've spent a lot of time on so far. What we've been looking at are patient-derived tumours. So these are tumours that we create in model systems from patient material. And remarkably, what we've found is they're very, very heterogeneous in the way they look in this imaging technique. And we're really trying to understand at the moment what that means, what it means for treatment, what it means for outcome, for example. So that's really been our focus. That's what I was talking about here. This technique we're about to take to the clinic later this year, early next year, and our focus there will be on early treatment monitoring. So in patients, for example, in breast cancer patients, where they're getting treatment and asking the question, are we seeing any treatment very early, are we seeing an effective treatment very early on? What we hope this means is that you can target therapy more effectively at those patients that were going to respond to a particular drug. So there are choices now to be made in treatment. And what we hope is that this will allow that treatment to be more effectively targeted at those patients that are going to respond. So if a patient's not responding, maybe you can change much earlier during the treatment process. So conventionally, the way we assess treatment response is to see whether the tumour shrinks, but that can take many weeks or even months before that becomes apparent. The methods that we're developing could tell you within 24, 48 hours whether anything's actually happening. We're developing other methods which are a much earlier stage of development. For example, we're looking at methods to image the surface of cells and how they change. This is a long way from clinical application, but we're developing new methods of imaging sugars on the surface of cells, which I find really fascinating and an interesting approach.